Hello everyone, in this video we're going to talk about the difference between allopatric and sympatric speciation, a conversation we already started last week, but we're going to continue and talk more about the sympatric speciation. In addition to this video, you should be completing a little bit more questions on your AP Classroom portal on evolution, a little more practice on this topic. We're getting towards the end of our learning of evolution, so make sure you get as much practice as you can. In addition to that, you're also going to be reading our curriculum, the course and exam description for homework tonight. You're going to read and annotate the entire packet and then make a note of, make a star or something, next to any part that we haven't already talked about in class. We'll address those problems in the near future of our classes, okay? So today we're going to talk about sympatric speciation and allopatric speciation. As a reminder, in allopatric speciation, there has to be a geographic isolation between two populations, and that restricts the gene flow, right? So what we mean by that is because two populations are geographically separated, they cannot interact, and they cannot even attempt to mate because they simply won't be able to see each other. And then reproductive isolation may then arise as a byproduct of genetic changes resulting from diversion, natural selection, genetic drift, or sexual selection. This is a lot of words. What do we mean by that? So what we mean is that when two populations are geographically separated, they're going to accumulate their individual mutations and natural selection and genetic drift, sexual selection. All of these factors will contribute to making the two populations more and more different from each other, right? That as a result, due to all these differences, eventually there could be a reproductive isolation as the end product. So as a reminder, what is reproductive isolation? Reproductive isolation talks about two different populations. They cannot interbreed and produce a viable fertile offspring, right? If a viable fertile offspring cannot be produced, we're saying that these two are two different species based on the biological species concept. So it's important for you to remember reproductive isolation is a result. It's not a precursor. We talk a lot about reproductive isolation as, you know, an evidence for us to know what are two different species, but reproductive isolation is really a result of an accumulation of different mutations, selections, and all of that make two different populations so different that they become reproductively isolated. Hope that makes sense. So even if contact is restored between the two populations, interbreeding is prevented. Even if these two populations eventually come in contact, they still cannot produce a viable fertile offspring because they're too different by this point. Okay, so in sympatric speciation, however, there is no reproductive barrier. We have two populations. Oh, what did I say? There is no geographical barrier is what I meant. So we have two populations that are not physically separated from each other. They can still meet each other, but they are either not going to attempt to mate, or even if they mate, they're going to have this reproductive barrier that prevent the two populations from forming a viable fertile offspring. So sympatric speciation can result from polyploidy, right? Remember last Friday we learned about how these plants, right, they have a certain number of chromosomes, and when the numbers of chromosomes come together and they form this zygote, the meiosis process cannot keep on going with a certain number of chromosomes. So if you don't remember this, go back and look at your notes or talk to your classmate. We also have natural selection, right? Natural selection in one area, even if two populations are in the same area, these two populations could have different habitat, different niche, that they are still going through natural selection separate from each other. And lastly, we have sexual selection. Remember the fish that we looked at? Even though the two populations of fish are in the same area, because they develop this preference for the looks of their mates, the sexual selection process eventually also left enough differences between the two populations that they became two different species. So let's continue on. We have hybrid zones. So hybrid zones is an area where hybrids are made, right? So if you have two populations that are two different species, they could come into contact at certain places, and we call that a hybrid zone. If a viable hybrid can be made. This hybrid might not be fertile, this hybrid might not live very long, but a hybrid is made. So a hybrid zone is a region in which a number of different species mate and produce hybrid. Like I just said, hybrids are the result of mating between two species with incomplete reproductive barriers. So what do we mean by that? If you think about horses and donkeys, that's an incomplete reproductive barrier. They're very different from each other, but they're not different enough, or they're different not in the way that they can't produce an offspring at all. So hybrids can be made in these hybrid zones due to incomplete reproductive barriers. A hybrid zone can also occur in a single band where adjacent species mate, right? So if you think about two geographical areas, if they meet along one line, sort of, then that's a very narrow hybrid zone. But you can also have patches of hybrid zones and individual species, as this next bullet point talks about. And then lastly, we have hybrids often have reduced fitness compared with parent species. And we're missing a little part, so I'm just going to read it to you. It says increased rates of embryonic mortality and morphological abnormalities, right? So hybrids can be made, but this hybrid might not be able to survive for very long, right? So the chance of this hybrid actually surviving and staying alive till adulthood, it might not be very high, right? And there could also be morphological abnormalities to this hybrid. Even though this hybrid is made, there could be certain aspects of the hybrid that is abnormal. And also a hybrid is very often infertile. So moving on to the next page, we can see that we have these two areas or two species of toads, and it's forming this hybrid zone along the two regions. So take a look at this picture right here. So this picture, this graph shows you some sort of data that you can read. So what you see is that along the x-axis, it's a little hard to see, it says distance from hybrid zone center. Okay, we have the hybrid zone center of the hybrid zone, and then we have the yellow-bellied toad range, which is over here, and we have the fire-bellied toad range, which is over here. So what you see is that the y-axis is the frequency of this. Which one is this? This y-axis is one of the species of a specific allele, right? So that's the yellow-bellied toad specific allele. So this particular allele, most of the yellow-bellied toad has it, most of the fire-bellied toad don't have it, but within the hybrid zone, you see this decreasing in the allele frequency of this particular gene. So what we mean by that is, as these two species interbreed in this hybrid zone, right, they're mixing up the genes that you have this decreasing in this one allele. Hope that makes sense to you. We have hybrid zones and environmental change. So we have a hybrid zone, right? But this hybrid zone might not stay the same, and actually won't stay the same for extended period of time. This hybrid zone can move around because animals, if this hybrid zone is caused by animals move around, right? They're not going to stay in the same area, so this hybrid zone can move. So we have changing environmental conditions can result in relocation of exhibiting hybrid zones or the production of novel hybrid zones, right? This hybrid zone can move in a certain direction, or you can even have a new hybrid zone over here, for example. And then the breeding between the hybrid and parent species can result in the transfer of allele, right? If you think about it, we're mixing up the gene pools, we're transferring the alleles from one parent species to another, which is also what this graph was trying to show you. All right, continue on. We have this little hybrid zone, right? We have these chickadees. So this hybrid zone actually moved north as a result of global warming. So we have these chickadees that live generally in a colder area, and we have the chickadees, I don't know why I'm saying it like that. We have these birds living generally in the southern region where in warmer weather, but this whole hybrid zone have actually moved north as a result of global warming. Another example are these little flying squirrels. They have this hybrid zone, and we have an overlap of actually, in the past, there is no hybridome between these two types of flying squirrels, but now there is. Okay, moving on. So hybridomes can change over time. So there are three possible results of a hybridome. We have reinforcement, fusion, and stability based on how fit the hybrid is, right? So let's take a look at these three outcomes. We have our gene flow, right? We have our original parent species, that is the common ancestor of both species. And then due to some sort of barrier, whether it's a geographical barrier or, all those other kinds we've learned, behavioral isolation, all of that, the gene flow is cut off. These two species are no longer, or these two populations are no longer breeding and mixing up their genes as often. So then they go through their own, again, mutations, natural selection, sexual selection, genetic drift. So now they become so different that they are now two different species. However, if they do come in contact, there could be a little hybridome where very few individuals of hybrids compared to the population, the individual populations, we have these hybrids that can be made. But there could be three outcomes: reinforcement, fusion, or stability. So what do we mean by that? A reinforcement is that if a hybrid is not very likely to survive, or even if it survives, as we said before, there might be some sort of physiological abnormalities, or this offspring might not be fertile. In that case, we could strengthen the reproductive barriers. It makes these two species more and more different because the hybrids are not very likely to survive. So the hybrids are being selected against, and the regular two different species are being selected for. Or if the hybrid is likely to survive, right? If the hybrid is viable and fertile very often, then there could be a weakening of the reproductive barrier. We're kind of selecting for the hybrids because they're fine, right? So as a result, we could have the two species being fused back together into the same species because you're weakening the reproductive barrier. We're having the two species constantly mating and mixing up the gene pools. Lastly, we can have stability if this hybrid is neither selected for or against. This hybrid is okay, is kind of in between, is neither dying a whole lot or surviving a whole lot. Then we could have these two species just staying constant. They're not becoming more and more different from each other or more similar to each other. So we have, I apologize for my lack of fluency. I've been up since 4 a.m. It's not great. So reinforcement, we have strengthening reproductive barriers, right? We said when hybrids are less fit than the parent species. So these hybrids are not surviving a whole lot, or even if they survived, they're not producing the F2 generations, then we're selecting against the hybrids. So as a result, the reproductive barriers can select even more for the prezygotic barriers. Over time, the rate of hybridization decreases, right? And then the two different species are staying more and more different from each other. So when reinforcement occurs, reproductive barriers should be stronger for sympatric than allopatric species. So if you think about it, if we have two different species, they can mix up their gene pools, but they still are not. That is a pretty big deal, right? It's as if you can talk to the person next to you, but you choose not to talk to this person ever. You must really hate each other kind of situation, right? If you have two species that can potentially breed with each other, they're in the same area, but they're not able to breed, that means they must have a really strong reproductive barrier. The next one is called fusion. So if hybrids are less fit than the parents, and a lot of times they don't survive, then gene flow, and if gene flow is great enough, right? If the two different species are keep on breeding with each other and their hybrids are very fit, then the reproductive barriers can be weakened, and then the pair species can form back together into the same species. So as an example, this one's really good. As an example, we have these two types of cichlids that we looked at last week. Remember, we had the two types of fish that have their own sexual selection, where one is selecting a certain pattern of fish and the other one is selecting a different pattern of fish. Those are two different species. However, due to pollution, the female fish cannot see so well. So now they're just mating with all the fish because they can't see which type of fish they're looking at, right? They can't actually choose the mate that they actually want. So now they're just mating with all kinds of fish. So now we have more of a hybrid of the two types of fish, and those hybrids are being formed a whole lot, that these two species of fish are no longer the same species of fish. No, are no longer two different species of fish. They're now the same species of fish because you're mixing up the gene pool so much that they can continuously produce viable fertile offsprings. Lastly, we have stability. So in this case, if we have extensive gene flow from the outside of the hybrid zone, basically the idea is if this hybrid is neither super fit or really not fit, then we can have this reproductive isolation between the two species, but then there's going to be an ongoing hybridization of the two zones, of the two species. I'm sorry. So lastly, we want to think about how fast can speciation events occur, right? As you should already know, based on your common sense and what we have learned in evolution, speciation can happen within a short amount of time or a long period of time due to the accumulation of, who knows, different numbers of genes, right? It really depends on what kind of species we're looking at. Sometimes you can only accumulate several differences, and that could lead to an entire new species, but other times you need to accumulate a lot more differences to allow the two populations to become two different species. So how long does it take? We really don't know. It really depends, right? However, we can look at the rate of speciation based on the fossil record, for example, right? You can do radioactive dating and see how long did it take going from this one species to the next by looking at the fossils. We can look at morphological data. You can look at the appearance of the species and try to measure how long did it take for this one species to become so different over time. And then lastly, we have molecular data. You can look at the DNAs and the proteins and see how long did it take for us to accumulate this many differences to allow these two populations to become two different species. Okay, we're gonna skip this part. Okay, so when you look at the fossil records, right, you can, what we see, okay, this part is really important. What we see is that instead of having a gradual change from one parent species into two completely different species, that's not actually what happens. What we see in the fossils is that species generally persist unchanged for a really long period of time, and then they disappear, or they reappear as a new species very quickly. So we call that punctuated equilibrium, or punctuated equilibrium. So that tells us we have a long period of time of unchange, and then it's a very fast period of time of change. This is almost like the chemistry curves that you've seen before, you know, when we have water remaining in the liquid state for a really long time, and then it becomes gas really quickly, and then it stays as gas for a really long time. Remember in chemistry when you see a curve like this, possibly? Okay, so the punctuated equilibrium model contrasts with the gradual change in a species over time. So that's called gradualism. The gradualism is incorrect. Punctuated equilibrium is more like what actually happens. So how does that work? So gradualism is when, again, one species being changing into two different species, but this is a very gradual process. It takes place over time, and it's a gradual change of the physical characteristics and the DNA of the two species. However, what really happens is that this one parent species is going to remain unchanged for a really long period of time, and due to some sort of selection process, whether it's natural selection or genetic drift or sexual selection, then there's going to be this very quick change from this one species to the two different species. Quick as in not 1 minute quick, but relatively quick period of time. And then all of a sudden you go from one species to two different species, and they remain as its own species for a really long time before another speciation event could happen. This next one is called speciation rates, right? As we've talked about, the rate of speciation, how fast can a speciation event happen, really depends. It could be a short period of time, it could be a really long period of time, it really depends. Okay, so we're gonna skip this experiment. Sometimes it could take about 4,000 years to 40 million years, with an average of 6.5 million years. This is just to give you a sense of how long speciation events can take place. It really depends. So speciation occurs only after gene flow between population is interrupted, right? Not after a given amount of time. So time doesn't guarantee new species, right? It's really can the gene flow, or does the gene flow get cut off? Whether is gene flow being cut off in the sense of Sympatric, where the two populations are at the same place, but then they stop mating with each other and they're cutting off this gene flow, or in terms of Allopatric, they're completely separate from each other, so then they are cutting off the gene flow as well. Studying the genetics of, okay. Lastly, we want to talk about how many genes influence the formation of new species, right? How much difference do you have to accumulate between the two species, before the two species actually become two different species? So again, it depends. Sometimes it could just be one gene, right? In this case, we have these snails, and this one gene affects the direction of the spiral and where it's you know, how they can mate, right? So if their shell is spiraling in another direction, and that influences where the reproductive organ is, then they're not going to be able to mate, okay? But other times there could be more than one gene, or even way more than that, changes before the two populations can become two different species. Lastly, this is our last slide. I know this is a little dreadful, I'm so sorry. It would be much better if I was actually there, but I'm not. I appreciate your patience with going through this entire PowerPoint with me. So this last slide, from speciation to macroevolution. Macroevolution is the sense of having actual speciation, right? But we said you need to think about speciation is not the only thing that happens. Extinction is also something that happens, and it's very important for evolution as well. So you need to remember both speciation and extinction contribute to macroevolution, and microevolution is simply the change in allele frequency, which is something that happens every single day. But that microevolution doesn't always lead to macroevolution, but they're both evolution. It's just on different scales, and we can look at both of them at the same time.